In the previous chapter, we explored the host-parasite relationship with an emphasis on the role of the microorganism in the disease. In the next couple of chapters, we're going to be examining the other side of the relationship, that of the host defending itself against microorganisms. This figure is an overview of all the host defenses. Of course, at this moment, it probably doesn't make a whole lot of sense to you, but it will make a really good summary slide for all that you're going to learn in the next couple of chapters. Host defenses are broken into two categories: the non-specific or innate defenses. These are the defenses that the body's ready with, regardless of whether you've had any exposure to this pathogen before. And then the specific or acquired defenses are those ones that require some previous experience and memory cells to remember the exposure. There are three lines of defense: the first, second, and third lines. In the first line of defense, we're dealing with barriers that block portals of entry. And in the second line of defense, we're dealing with the internalized system of protective cells and fluid. The first and second line of defense are both non-specific defenses. This is where the body is always on guard. The defenses don't improve with repeated exposure. It involves things like barriers and inflammation and phagocytosis. This is the content of this chapter. The third line of defense, however, is where we see highly specific, acquired, long-term immunity. This is where the lymphocyte will encounter invaders and gain memory for future invasions. That'll be the content of the next chapter. The figure over here on your right summarizes all of these defenses. Host defenses are broken into the non-specific and specific responses. The non-specific involves the first and second line, whereas the specific defense is all the third line of defense. We're going to get into a number of physical barriers, chemical barriers, and genetic components involved in the first line of defense, as well as inflammatory response, interferons, phagocytosis, and then the complement system involved in the second line of defense. In the third line of defense in the next chapter is where we'll see all this specific immunity, some that's naturally acquired and others that are artificially acquired. Let's begin by looking at the barriers, the first line of defense. These are inborn, non-specific defenses, and they're divided into three basic groups. We'll consider the physical or anatomical barriers, the chemical barriers, as well as some genetic barriers. So, beginning with the physical or anatomical barriers, we can look at the thick skin. Our skin, its outer layer, is composed of stratum corneum, which is a highly impervious, super compacted layers of stratified squamous epithelium that's interlaced with a large amount of keratin, which gives it this impervious, waterproof quality. Few pathogens can penetrate unbroken skin. The skin as a barrier between our inner bodies and the microbes on our outside is often overlooked as a barrier. But really, when you consider it, it's a pretty obvious one. In addition to separating our inner bodies from the microbes on the outside of the bodies, we see that the outer layers of the skin are sloughed off or desquamated. As the skin is sloughed off, it takes away many microbes. In areas where we don't have skin and we connect with the outside, there are mucous membranes. The mucus layer on these membranes impedes entry and attachment of microbes. In our lungs, we have cilia, or little hair-like structures that you can see on the cells here. These cells, along with mucus, act to push pathogens up and out of the lungs. Literally, these hair-like cells beat in an upwards action in order to move mucus up through the respiratory tract. Most of this is then swallowed and destroyed when it reaches the stomach. This system of mucus and cilia working to move substances up and out of the lungs is called the ciliary escalator. Smoking severely damages these cilia and prevents them from acting in this escalator, and thus we see that smokers have to cough more in order to remove the mucus that begins to pool in their lungs. Another external barrier are all sorts of secretions. These secretions have flushing effects, like our sweat glands flush our skin, removing microbes along the way. We have tears that are constantly released across our eye membranes, the process of lacrimation. This is tearing and blinking that flushes our eyes. Our saliva carries microbes constantly from our mouth down into our stomach. The high acidity of the stomach kills most microbes. Then, of course, vomiting and defecation. If you've ever had food poisoning, you know these very well. They're there for a purpose. The body identifies that there are microbes invading. As soon as they're recognized, we'll begin to vomit and defecate, move things through the system fast so we can get rid of them. We also see, again, along the ciliary escalator idea, there are mucus secretions in the respiratory system. Like if you have allergies or colds, you'll see an increase in the production of mucus, constant runny nose. This is of purpose so that we can flush microbes that may have invaded the mucous membranes of our respiratory system. Sneezing and coughing also act to remove some of these secretions to get rid of invading microbes. We also know that there's a constant little drip of urine through the ureters, through the urinary system, constantly rinsing potential microbial infections. Vaginal secretions act in a similar way. Constant flow of these secretions acts to continually cleanse the vaginal environment. Now, although the resident biota aren't technically a barrier themselves, we've covered the idea that their presence also creates unfavorable conditions to invaders. They definitely want to stake out their own homes, and they might create an unfavorable chemical environment. Now, on this note, there's a great deal of research that has highlighted the importance of gut microbiota and how it impacts the development of both nonspecific and specific immunity. They say that the presence of the biota, our natural biota, trains the host defenses. Evidence suggests that if we interrupt this process, say with antibiotic treatments that remove all of our resident gut biota, it will lead to immunologic disturbances in the gut. Some scientists believe that the inflammatory bowel disease that we're seeing so prevalently these days is a result of the overzealous attempts to free our environment of microbes and overtreat ourselves with antibiotics. So this creates an ill-trained gut defense system that then inappropriately responds to very common natural commensal biota that should be in our gut. And this is a really interesting thing to think about. It all seems to make sense to me. As you can see, before microbes even have an opportunity to be exposed to our second and third line of defenses, there's a whole array of barriers at the body's surface. Next, let's take a look at some of the chemical barriers. We'll see sebaceous secretions. These secretions from glands in our skin have an antimicrobial effect. Also, glands in our eyelids, the meibomian glands, have antimicrobial secretions. In our tears is an enzyme called lysozyme. This enzyme hydrolyzes peptidoglycan that's seen in cell walls of bacteria. We also see that lactic acid and electrolyte concentrations are very high in sweat. This adds to the skin's acidic pH as well as fatty acid content, both of which create an antimicrobial effect. Next, once any of the secretions that have been used to flush tissues reach the stomach, it has a highly acidic environment because of the hydrochloric acid content. This acidity kills any of these swallowed pathogens. Digestive juices and bile in the intestine also act to break down microbial invaders. Then, when we look at our reproductive organs, semen contains an antimicrobial chemical so that it prevents microbial infections. In addition, the vagina has a very acidic pH. This, we've already explored, is maintained by a balance of the resident biota, yeasts, and bacteria. Finally, when we consider our barrier defenses, there are some differences in genetic susceptibility. Some pathogens have a great specificity to one host species. That is, some hosts are genetically immune to the diseases of other hosts. For example, you won't see a human get cat distemper, and cats certainly can't get mumps from humans. This specificity is particularly true of viruses because, as we've already explored, they need a specific host receptor to gain entry to those host cells. Now, we've also explored zoonotic infections. Naturally, zoonotic infections, by their definition, are able to cross over the species barriers, so they are less specific. Now, there are also genetic differences in the host susceptibility. These could come from mutations in genes that code for components of the defense system, like T cells and their receptors, the complement proteins, and the cytokines, all of which we'll be exploring later on in these chapters. So to go back to this figure, which we'll continue to come back to, we're looking at our innate or non-specific defenses. We've considered the first line of defense, where we had physical barriers, chemical barriers, and genetic components that prevented entry of microbes. This first line of defense is the surface protection layer. It's composed of anatomical and physiological barriers that keep microbes from penetrating the sterile body compartments. So far so good, right? We've now covered the first line of defense. Before we move on to looking at the details of the second and third lines of defense, let's have an overview of what those systems do in general, because from here on out things get slightly more complex. Now, although in this course we're considering infectious microbial agents, remember that immunology is a large consideration in many other fields. For example, in the study of cancer and allergies, immunology is a huge component. Immunology involves the study of all features of the body's second and third lines of defenses. So when we refer to the immune system, we're looking at second and third line defenses. Healthy functioning immune systems are responsible for surveillance of the body, recognition of foreign material, and then destruction of entities that are deemed to be foreign. So in essence, we're into search, recognize, and destroy. And the destruction occurs mainly through white blood cells. They roam around the body continually searching for potential pathogens. These white blood cells are trained to distinguish self from non-self cells. They evaluate other cells by examining markers on their surfaces. These markers are composed of proteins and sugars, or both. And essentially, they're the equivalent of facial characteristics in humans. So white blood cells run around the body. They're equipped with a very sensitive touch. And as they sort through the tissues, they feel the surface markers, which help them determine whether that cell is self or non-self. If they're recognizing self, then no response occurs. However, if the white blood cell contacts a pathogen, recognizes it, then it will certainly destroy that pathogen. So the second line of defense is still a non-specific or innate line of defense. It involves things like inflammation, interferons, which we'll explore soon, the process of phagocytosis, and the complement system. The third line of defense, which we'll cover in a following chapter, is an acquired and specific defense system. It involves antibodies, beta cells, T cells, and memory of the pathogen present. So in future exposures, all the equipment is lined up to destroy that pathogen specifically. Now you might ask, what about all the proteins that are not self, but they're also harmless, like the food we eat or a resident biota? Well, generally, those are recognized as not a threat, so they're left alone. This is the issue with increasing numbers of allergies in the immune system. Probably again due to the overuse of antibiotics. We've kind of screwed up the system so that our body is not able to recognize a non-threatening protein or microbe. And thus, the immune response fires up inappropriately.